So students, please type in the chat box if everything is fine so that we can start the session. If my voice is fine, if screen is fine, streaming is fine, then we can start. Because somehow I cannot read your chat box. Cannot. Yeah, sorry, it was earlier due to some strange thing. It was not able to do so. So let's start the class then. Good, good, good. So keep chatting with me so that I could be in touch with you. Otherwise, it will not be so useful for me to keep speaking to myself. So I will keep asking you things. We will keep interacting with each other via chatting so that we can have a good, healthy class. Yeah. So if you have any questions or anything that you have any query, you can type me in the chat box or type me later on also on the Telegram. So I will try my best to answer your questions of any kind. And yeah, so let's start the class. So I have shared this thing with you already, I guess, the objective. So we will start with the first objective, that is your nucleic acids and their structures, nucleic acid, how there is a genetic material, and yeah, types of DNA later. So basically, I want to give you for the moment a basic feel of basics of your molecular biology. What is it's all about, and what does it say at the end. So let's start. So if we starting with basically, as we have discussed today in this Zoom session, that what is molecular biology is, what we are made up of is we are made up of all ATCG, all these four nucleotide molecules, and because of that we are working and we're gonna study in core details how these ATCGs play a big role to make the central dogma of life in order to survive and make things done. So let's see one by one. So the eukaryotes, Just a second. Just a second, students. But can you see all these content that I'm showing you? Is it properly visible to you? I think so, yeah. So if can you read? Can you able to see this 80s, 70s written absent? Is this visible to all of you? Because there will be some tiny minute details will be coming. If they are not, you're not able to see them. So if they are blur or something is not properly for you, so it will be very shame for me to make the class happen. Try for some students, if it is blur, try to make it to 480p, then it will be fine. At 480p it's working very well without any problem. Good. So most of the students are fine with it now. Just who's having this problem, please make it to 480p in your mobile or in your laptop, so that it will be fine afterwards. Okay. So eukaryotes versus prokaryotes. So basically, the eukaryotes we know that belongs to us as a human beings. We plants, animals, fungi, they are all eukaryotes, and the prokaryotes are the one with the bacteria and cyanobacteria, the primitive one. They don't have any cell wall. They don't have plants have the cell wall so far. The prokaryotes do have also. Beyond that, meiosis is not happening in eukaryotes. In prokaryotes, it's absent, and the ribosome size is of 80s in eukaryotes, and the prokaryotes it's 70s. And all the membrane bound organelle, that is your nuclear membrane, endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, they are all present in the eukaryotes, where in the prokaryotes they are all absent. Then molecular biology, the main definition behind this. So molecular biology is a study of molecular underpinning of the process of replication, transcription and translation of the genetic material. Though I said this statement within maybe 3 seconds or in 5 seconds, but believe me, this statement plays a quite important role in order to understand the mechanism of the human body. And this took a lot of years to write this sentence also. So within this sentence, many research, many hard work, many Nobel Prize laureates, many findings were belong to it. So what does this statement again? I will repeat that. That is, molecular biology is the study of moleculars underpinning of the process of it includes the study of those molecules which deals with the process of your replication, transcription and translation of the genetic material. So here we can see that it includes, first of all, the DNA could replicate during the mitosis or meiosis process, and then further the DNA could be transferred to the messenger RNA, and then during the transcription process, and after the transcription, this messenger RNA with the help of your ribosomes and tRNA will produce your proteins in the process named called as translation. Yeah. So and beyond that, this molecular biology is quite interdisciplinary field in which most of your other fields are has to be interconnected with molecular biology, because this cannot work with the help of with each other. So if somebody wants to discuss with plant development and defense system, like these growth hormones, this defensive system, how the plant defense system work, So we use model system of Rhabdopsis thaliana. If somebody had worked in the plant field research, so Rhabdopsis thaliana used as a model plant in order to find any disease resistance genes or transgenic plants you want to make, or you are looking for extremophiles, the bacteria which could survive in the extreme temperatures or extreme pH or extreme conditions, and we can extract their genes in order to create some drugs for human body, so that we can also survive the extreme temperatures with those. So enzymatic production, like various enzymes are very playing important role as a biocatalyst in our human body also, and also from the industrial process. Whether it's a food industry or a medicine industry or pharma industry, everywhere enzyme is playing a big role there. Then comes structural molecular biology. There's also, if you want to produce any drug or any inhibitor against some target thing, or you're producing even this vaccine against this COVID-19 virus. So all to understand that, we need to understand the molecular biology of this virus, which are the spikes proteins that are coming into our human body and getting attached to our larynx and pharynx and making our whole breathing system blocked. So it will be very nice to understand the structure, molecular biology of various structures. And then genomics, as we know, only 2% of human body, 2% of your total genes, they are corresponding to the protein. They are translating to the protein. Only 2, or even less than 2%. Rest 99% is of no use in your whole genomes. All genes have no use in that. So you can see how little amount of gene has such an importance, and the rest genes are still undiscovered. What the use are, we are still unknown. Then there is also molecular entomology, so that we can extract from the various insects like bioluminescence insects. Jugnu, we say jugnu hota hai na, तो जुगनू से हम we can create, we can extract that gene that which make them lighten up in the dark, and we can create some other dyes or some other workout, fireflies. From fireflies we can create some dyes in order to see them, our cells also, for the molecular biology perspective. So we can see that the molecular biology itself has important role in the cells, plants, microorganisms, molecules, populations, and animals. It has a wide variety of importance, and this will be a big field in upcoming days. At the moment, the field that is really helping the molecular biologist is the computational biology, actually, the bioinformaticians, because these people, they have created such big algorithms, such very nice techniques with the help of artificial intelligence and machine learning, Python and all these new languages. So they can create really good formulas in order to predict the structural, for the drug discovery, for molecular docking, and all these things which as an organic chemist, when once they go into the lab and they wanted to discover one drug, so for them they took like 10 years to do so. But with the help of bioinformatician, you can predict these structures within 2 weeks, and money is very less efforts, very less frustration, and yeah, and things are also bringing up front for the society. So the many big pharma companies are really paying to these to the IT bioinformaticians for the upcoming future. So that was some basics about molecular biology, a basic push that what is it all about. So the field that overlaps other areas of like biology and chemistry, particularly genetics and chemistry. So some students were saying they are interested in the genetics in upcoming days, or going for forensic science or something like that. So something related to all these fields, they are overlapping as we have discussed. So a good to understand the good interaction between your DNA, RNA, protein biosynthesis, how they are regulated, that will be very essential key things in order to understand the molecular biology. So further, in 1950s and 1960s, molecular biologists have learned to characterize, isolate, and manipulate the molecular components of cells. So we can extract the DNA and then get to know about what are the various sequences are. We can create a lot of multiple copies with the help of PCR, and we can also from RNA, after production of RNA from DNA, we can create complementary DNA also. Beyond that, then after the mRNA is being produced, we can produce various proteins and make enzymatic type of molecules that are required for the functions. So what are the basics components that involve in the molecular biology? That is your DNA, RNA and protein. That is your DNA, RNA and your protein. So how these components are important, why they are required, is just because of the central dogma of the life. That's the central to the life actually. So from your DNA, your mRNA is being produced. From double helix, a single double helix mixture of RNA is being produced. Then from this RNA, your protein is being produced having a quaternary structure. So we can see gene as a unit of heredity, as in which has segments that carries genetic informations that are called genes. And it's normally a stretch of DNA that codes for a type of protein for an RNA chain that functions in an organism. In order to explain you this diagram, let me take you to the this diagram. So here it's been shown that how our tiny DNA of 2 nanometer of diameter has been intermingled to a chromosome of 1400 nanometer of size. So how things are interlinked with each other and making a sense at end of the day. Yeah. So here we can see. So DNA double helix structure, we can see it's of 2 nanometer. Yeah, this is a 2 nanometer of structure, double helix structure with all your nucleotides, and then this is the simplest level of chromatin in a double stranded helical structure of DNA, very simple form. So we will see now the next part. The DNA has been complexed with the histones to form nucleosomes. So these are your nucleosomes being covered by this DNA, and the inside is the nucleosome core of 8 histone molecules. And on that side, what we can see is histone 1, which act as a clipping agent in order to cover your whole covering of this beads. So these are also called as beads on a string. Yeah, these are the beads on a one string are being attached. So this is called bead on a string structure. And then each nucleosome consists of 8 histone proteins, around which there is a wrapping of your DNA about 1.65 times. Then later on, these chromosomes also consist of nucleosome plus with H1 protein, as we have discussed. So inside there is a H3, H4, H2A and H2B. So H2A and H2B, they are they are coming together. Then H3 and H4, they are in the tetramer. So in total, it's an octamer actually. So 8 histones proteins are present in this circular form, in the like a Costco ball. And on the side there's a histone 1 is being present. Yeah, and then these beads on a string structure, they get evolved further in order to produce nucleosomes folds up the produce of 30 nanometer fiber, so that the length or the diameter of these strings is now 30 nanometer from 2 nanometer. And these strings, they keep wrapping up in order to form loops or average length of 300 nanometer. Then from 300 nanometer, they start to crisscross with each other, make a zigzag like this thing, making up to a 700 nanometers. And then they make the sister chromatids of your chromosomes, your shorter and the longer chromatids. And the tight coiling of 250 nanometers of fibers produce the chromatids of a chromosome in this way. So I will repeat this thing because this might be not clear at the first one go. So what I said, that there is a DNA double helix structure of 2 nanometer. Yeah, which is the simplest level chromatin with a double-stranded helical structure of DNA. Then they got coiled around the nucleosome, core of 8 histone molecules, and then there is a one, yeah, histone 1 is being present on the outside, and these chromatosomes uh, are playing role, and this together thing, the 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 DNA that is wrapping around your histones, that is known as a chromatosome altogether. And this is length of 11 nanometer, and this keep going on in order to produce these rings of 30 nanometer, and then this form around 300 nanometer in the length, then the zigzag sections of 250 nanometer, and then they become the sister chromatids of your chromosomes. So that was one important diagram. So I should share this at the moment because so this diagram is just showing the summary of that we have discussed. The genes consist of these histones body, and they are being carried out over the things there. Just a second, bache. Yes. Yeah. Yes. Yeah, ma'am. Yes, ma'am. Actually, I'm teaching. Actually. Yeah, yeah, yeah. Vedanta course, maybe ma'am, some of my friend might have done it from my email ID also, but no, I haven't done anything like that. Yeah, if the yeah, you too, ma'am. Thank you so much, ma'am. Yeah, ma'am. Actually, I'm taking a class at the moment. Maybe I'm in touch with you later. Yeah. Yeah. Okay. 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 So there was one call from that I have enrolled for some courses. Uh, so yeah. So we were talking about these genetic material, how things are working out. Cool. So you can see this chromosome, it will have these dark bands, and they correspond to gene 1 and gene 2, and there are some like white bands, it doesn't correspond to the coded part. So that's how your gene as a unit of heredity works out. Then comes your deoxyribonucleic acids, which is a nucleic acid that contain the genetic instructions used in the development and functionings of all knowing living organisms and some viruses. So it's a set of blueprints needed to construct other components of cells such as proteins, RNA, and molecules. So we can see it's a double helix structure, very nicely done with the constructed with the help of nucleotide pairs. On the backbone we are having our sugar phosphate backbone chain. So we can see these structures. Sorry for the picture here, a bit blurry picture. So these are the two long strands that make the shape of double helix structure here. So like this, ATCG, ATCG, and the diameter is around 2 nanometers, and the distance between each base pair is around 0.34 nanometer, and the distance between your major groove and minor groove together in the length is 3.4 nanometer. Yeah, and further that how this pairing of your hydrogen, this double helix structure is maintained in such a nice order is all because of the double and triple bonds between your nitrogenous base pairs. So here we can see the hydrogen bond between T and A and T is a double bond, whereas between G and C is a triple bond. So it's been continuing, and in between we are having this sugar, your deoxyribose sugar being attached with another deoxyribose sugar with the help of your phosphate groups. And that's how the 5' carbon on the top on the left and 3' carbon at the bottom. So 5' to 3' end, our double helix structure is being made. On the other hand side, on the right hand side, 5' end to 3' end is the other structure on the right hand side is being made. So that's why it's called as an anti-parallel structure. And so chemically, DNA consists of two long polymers of simple units called nucleotides with backbones made up of base, sugars and phosphate groups. Then what is the difference between your nucleosides and nucleosides? Basically, nucleosides is the one which has only sugar and your nucleotide base pair, your nitrogenous base pair, sorry, whereas the phosphate plus sugar plus base together they makes nucleotide. So that's the major difference, that in nucleotide phosphate is present and in nucleoside phosphate is not present. Then comes your bases, various nitrogenous base pairs, that is your purines and pyrimidines. So there is always a bonding between your purines and pyrimidines, that is A will pair with T with a double bond and G will pair with the C with a triple bond. And in RNA sometimes so in RNA sometimes we are having what you say, A is being transferred T is transferred to the uracil. Instead of thymine, uracil is coming up. So adenine, guanine, cytosine, thymine and uracil. So all these base pairs, they are playing big role there. So adenine has, so your purines are actually fusion of 5 and 6 membered heterocyclic compounds. Whereas the other one, cytosine, thymine and uracil, these are three different cytosine, thymine and uracil together, they make pyrimidine, single base pairs. And the basic difference between thymine and uracil is this methyl group. There is an absence of methyl group on that carbon. Just because of that, thymine is different from your uracil. So that's how the major difference between your purines and pyrimidines. And as we have discussed, that fifth pyrimidine base, uracil, this takes place of thymine in RNA, and just mainly difference of the methyl group in it. Then the pairing between A and T is double bond, and also A and is double bond, whereas the G and C is triple bond here. Further, the double bond between adenine and thymine is being made like this. So you have a nitrogen base pair on the top of the both amino the carbon, this hexagon 6-membered ring chain, and they're making these hydrogen bonds. Whereas in the guanine and cytosine, they're making a very nice triple bonds like this. And further, the major difference between your DNA and RNA is the sugar, with two deoxyribose sugars in DNA being replaced by the alternative pentose sugars ribose in RNA. So you can see the second carbon atom, it doesn't have the hydroxyl group. That's why it's called as deoxyribose. Whereas in the RNA, it's present, the OH group is present, and it's called as ribose sugar. So this one is present in RNA, this sugar, and in DNA, this sugar is present. That's why it's called deoxyribose sugar, and RNA is called as ribose sugar, ribose nucleotide acid. So this is the chain that we see, the small groove that we see, it's known as a minor groove, and the major groove is the one with the larger one. The DNA chain, the diameter as we have discussed around 2.5 nanometer, and the distance between each base pair is around 0.34 nanometer. So this coming sometimes in exams on the GATE and also NET exam, quite common. Then comes your ribonucleic acids, that is your RNA, which is a biologically important type of molecules that consist of long chains of nucleotide units. It also has nitrogenous base like same like DNA, ribose sugar and phosphate, but it doesn't have a continuous double helix structures. It's sometimes a single chain and then comes double chain and then again become single chain like we see in this diagram. So we are having these base pairs U, G, C, all these, and we have these hydrogen bonds being present, making between them. And yeah, so like this we are having this nice diagram of RNA. Then comes double-stranded RNAs. So double-stranded RNA is RNA with two complementary strands similar to DNA found in all cells. Mostly in the viruses it's single-stranded, but in the mostly in the normal cases RNA is single-stranded, but in the viruses it's a double-stranded. So this COVID, this spike proteins, and this is HIV virus actually, which is showing the RNA as a genetic material, and the protein code is present outside, having double-stranded RNA. So what are the types of RNA that is available in the human body? So if we see from the perspective as a major perspective, there are three types: messenger RNA, ribosomal RNA, and transfer RNA. And the abbreviations are mRNA, rRNA, tRNA. You might be aware of all these. And the functions is that messenger RNA codes for proteins, and ribosomal and tRNA is majorly used for the translation process. And once the translation is done, there is a post-translational modifications are happening, such as splicing, RNA processing, telomere synthesis. For the respective of them, your snRNA, yRNA, and telomerase RNA are being used. On the other hand side, there are also regulatory RNAs such as antisense RNA, which are used for your transcription, making mRNA stabilizations, and also to make mRNA degradations, which is happening in most of your organisms. So let's take one by one RNA, that how they are working. If we start from messenger RNA, so messenger RNA carries information about protein sequence to ribosomes, and these protein synthesis, they act as a factories in the cell actually. So it is coded that so every 3 nucleotides codon correspond to 1 amino acids. So here we can see this the DNA get open in the eukaryotic cells, and all the intron parts that are not required will be removed, and then we will have mainly these messenger RNA, which is produced from the coding strand, and there will be another non-coding strand which will be of no use. So we will have our product that we want as a messenger RNA as a डाकिया जो कि आगे जाएगा आपका. Then this messenger RNA is exported from nucleus to cytoplasm. So this statement itself is very, I would say, quite important term, that messenger RNA work when is done, that it's got transcribed with the help of DNA, and then it will bind, it will move from nucleus to the cytoplasm, and then bound to the ribosomes, and then translated into the corresponding proteins with the help of your tRNA. Yeah, so it is being bonded to ribosomes and translated to its corresponding proteins formed with the help of tRNA. So in prokaryotic cells which do not have nucleus and cytoplasm compartments, mRNA bind to ribosome where it is being transferred from RNA. So let's discuss this statement, that what we have been governed here, that which is showing the central dogma of the life, production of mRNA from DNA and then protein from mRNA in order to produce your successful proteins which will be useful for various functions. So on that note, I will take to you one video here. Just a second. Oh no. So Transcription and translation is done.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA.
Oops, sorry. I will start again.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
Okay, so let's choose a gene here.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So here we can see your DNA is being unwinded and we want to produce our messenger RNA. So corresponding, we want to give a try. So please give your comments. What we should do. What should we put in the answer? Yeah. So just type in the chat box what should be the answer for G. Shall I go for C, A, G or U? What type in the chat box, students? Will it be C, A, G or U? Is it G? G for G or G for C? It will be C. For A, it will be G or C or A. It will be U. For G, then it will be again C. For A, U. For T, it will be A. A. A is for U. C is for G. A is for U. C is for G. And T is for A. A is for U. C is for G. C is for G. So we will not forget this. Yeah, remember this for always. A is for U. So
When
like
the
this.
end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
Yeah, now let's give your answers on these topics with UUA, ACG, AGG, AU. Which one shall we go for it? UUA. So for translation, if our mRNA is coming out from the nucleus and it's being attached to the ribosome and also the transfer RNA, so which anticodon will work for it, and which amino acid will it be? Will it be valine, leucine, asparagine, or serine? UUA. Then it will be AAU actually. So it will be, answer will be leucine, or what else? Tell me. So it will be leucine. Then AGC. It will be UCG. It will be serine. Then AGC. For AGC, it will be UCG. That is your serine. So like this, it will continue. Now let's continue with another one. GCA. It will be CGU. AGU. It will be UCA. Serine. And then, so like this, it's continuing. Yeah.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, a proteasome, will now chop up other proteins for recycling. Proteins get sent to the proteasome when they have finished doing their jobs, when they are damaged, or when the cell determines that they have come from an outside invader, such as a virus. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So that was about transcription and translation working out within the cell. So I will share this nice videos link here over the Telegram group. So pin them, students, whosoever are present here. You can pin these things. There's a pinning option. Pin this, so you can come and watch them anytime of the day. So you can pin these things, the objectives, the links, and everything together. So let's continue. So we have discussed how your transfer RNA is playing role, which is a small chain of RNA chain of about 80 nucleotides that transfer the specific amino acids to the growing polypeptide chains at the ribosomal site of protein synthesis, and it has a site for amino acids attachments and anticodons regions for the codon recognitions, and it has a site that binds to specific sequence of the messenger RNA through hydrogen bonding. Then comes your ribosomal RNA. It's of two types, that is 70 and 80S, and the 70S is made up of 50S and 30S, whereas 80S is made up of your 60S and 40S. Yeah, so ribosomal RNA is a catalytic components of the ribosomes, and eukaryotic ribosomes contain 4 different ribosomal RNA molecules. That is your 18S, 5.8S, 28S, and 5S ribosomal RNA. And ribosomal RNA molecules are synthesized in the nucleolus, and in the cytoplasm, ribosomal RNA and protein combine to form nucleoprotein called ribosomes. And ribosomal RNA is extremely abundant and makes around about 80% of that L milligram per ml RNA found in the total typical eukaryotic cytoplasm. Now some differences between DNA and RNA. We have discussed both DNA and RNA, very basic about them. That is your RNA, it's a RNA nucleotide contains ribose sugar, whereas DNA contains deoxyribose sugar. RNA has base uracil and DNA has a base thymine. And the RNA on the other hand side is the presence of hydroxyl group at the second position of the ribose sugar, whereas the DNA lacks the hydroxyl group at the second position of the ribose sugar. And RNA is usually single stranded, whereas the DNA is usually double stranded. So in protein, also known as polypeptides, are made up of amino acids arranged. So next part is protein. We have discussed the DNA and RNA. Now comes the major part, that is your protein. So they are also known as polypeptides made up of amino acids arranged in a linear chain and folded into a globular form. So the sequence of amino acids in a protein is defined as a sequence of genes which is encoded in the genetic code. So there are around 20 standard amino acids. Like this, on the left hand side we can see cysteines and cysteines making these disulfide bridges, and their amine group is present at the end with the lysine, valine, phenylalanine, glycine, arginine, cysteine, glutamate, leucine, alanine, like this. So like you have this big structure, and they all make these disulfide between each other to make a quaternary structure here. And these proteins are present in our pulses, fish, egg, meat, red kidney pea, peanuts, chickpea, chicken breast. So all in these we having high amount of protein present. So how these DNA, RNA and proteins, they are playing role actually. So basically what they do is they do the replication, transcription and translation, and act as a central dogma of life. So DNA could replicate itself by themselves, and also could then further go for RNA, and from RNA the protein could produce. But remember, from DNA to protein this is never possible. Yeah, this is never possible. And also there is thing you can produce from your mRNA, you can produce complementary DNA. Yeah, this is also possible, reverse transcription. But yeah, but from DNA to protein is no way, no possible. But from RNA, DNA could go back, and yeah, and also from protein you cannot produce protein RNA. So these two things, these two things are not possible, but only from RNA to DNA. Oh, my battery is less. Is possible, and that is known as the central dogma of life. So it's quite important, the central dogma, how the central dogma works. It was given by the James Watson and Crick model. So in order to further take you into the world of videos of these, let me show you the basics of Yeah, what are DNA and genes? How they are differentiated between each other? It's a very small video about that.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order, but small differences in the sequence of the bases in our genes make each person unique.
So that's how these differences in our gene of 1 to 2% or even 0.1% make each us of different person here. That's how DNA genes are playing a major role in our body. So let's build a DNA molecule. Okay, they are not supporting it. Strange. Maybe we can see this same genes and different organisms like jellyfish having these colored dyes in them. We can use them, those dyes, in order to color our DNA or plants to see their cellular effects. We can use spider web silk proteins to put them in the yeast to increase the amount of spider silk for our better use. So we'll watch something about that now.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
I hope that was inspiring. This is the last one to discuss about proteins. We have discussed about DNA and GN, and we have discussed how same gene, different organisms, and a little bit about protein here.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, Nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
Yeah. So just go through this website. You will see many links available. This one. I hope my streaming is working fine. This one. And this one was not working. And this one. Okay, so now we continue with DNA replication, which is a basis for biological inheritance, which is a fundamental process that occurs in all living organisms in order to copy the DNA. So in this process of replication, each strand of your original stranded DNA molecule, which serve as a template for the reproduction of complementary strand. So there are two identical DNA molecules have been produced from single double-stranded DNA molecules at the end. Then you can see here how this DNA replication is done. First of all, you have your normal 5' to 3' uh, DNA molecule, and then it is being unwinded with the help of topoisomerase enzyme. This topoisomerase enzyme will unwind your DNA. Then the helicase enzyme will come and start to cut it from the center to separate all your nitrogenous base pairs which are bonded via hydrogen bond. It will start to break them. Then afterwards, your single strand binding proteins will come in order to separate between your each of your nitrogenous base pairs. Then there will be two strands will be made. One will be lagging strand, one will be leading strand. So in the leading strand, what will happen from the left with the help of DNA polymerase enzyme, with the help of your DNA polymerase enzyme, it will start to make new you can see this new DNA base pairs and started to work upon that. And then the DNA polymerase alpha, yeah, the DNA polymerase alpha, it will also come, and the bottom is the lambda delta, sorry, then on the top the alpha will play the role. But sometimes while doing so, some Okazaki fragments are being made. These are the fragments which are not being completely made, they are broken one, and these broken parts will be covered by your DNA ligase and RNA primer and DNA primase. So they will help us to control, to make them come up and join them together and make a smooth um, DNA pair. So we can see it's like a fork, replication fork. So we having this fork, for eating noodles or so. So it's become like a fork at the end, and that's why it's also called as replication fork. So remember three things in this. First is your topoisomerase will unwind your DNA and the helicase will start to break it up. Then there will be two strands, lagging strand and leading strands. And then basically your DNA polymerase alpha and lambda, they will start to produce your corresponding your nucleotides. And in sometimes there is Okazaki fragments. Okazaki name was given by the famous scientist Okazaki. Based on his research, this name was given, and then it's done. So that's the complete summary of your DNA replication. So in a cell, DNA replications begins at a specific locations in the genome called origins. An unwinding of DNA at the origins and synthesis of new strands form a replication fork. And in addition, DNA polymerase enzymes that synthesize the new DNA by adding nucleotides that are matched to the template strand. Then cellular proofreading is done with nearly perfect fidelity for DNA replication. Now comes transcription, that is a process by which you from DNA copy of your RNA is being made, and transcription is the first step, leading step for the gene expression. So you are having DNA giving rise to RNA with the process of transcription, but this could be also be reversible with the help of your reverse transcriptase enzyme and leads to the production of complementary DNA, cDNA. So during the transcription, basically DNA sequence is read by RNA polymerase, which produces complementary antiparallel RNA strands. So transcription results in an RNA complement that includes uracil instead of thymine. Then comes your transcription process. In the transcription, as we have discussed in details, that how your messenger RNA is being produced from your DNA with the complementary sequences. So during this stretch of DNA transcribed into an RNA molecule is called your transcription unit, which encodes at least one gene. If the gene transcribed encodes for a protein that results of transcription is known as messenger RNA, and this messenger RNA will be used to create that protein by the process of translation. And then alternatively, this transcribed gene may encode for either ribosomal RNA or tRNA and other components of proteins. And the DNA transcription unit encoded for proteins and regulatory sequences that directly regulate the synthesis of that protein. So here we can see the RNA polymerase, this is your promoter sequences. It's being copied to your DNA strand. This is a complementary strand, this is a copied DNA strand. And this RNA polymerase will unwind that thing, and then the self-complementary sequence will produce, and from that we will have our mRNA being produced, and our RNA polymerase will get removed. So in this way, our DNA is read from 3' to 5' end during transcription, but your RNA is created from 5' to 3' direction in this. And also during this RNA processing, remember that there are some exons and introns. So exons are the coding part, introns are the non-coding part. So during the transcription, your RNA splicing, your introns will be removed, and mainly your exons part will remain at the end, which will be used for RNA synthesis and processing. Then reverse transcription. So reverse transcribing viruses replicate the genomes by reverse transcribing the DNA copies from their RNA, and these DNA copies are then transcribed to new RNA, and the retrotransposons are also spread by copying DNA and RNA from one another. So we have HIV RNA here, yeah, and then they, with the help of reverse transcriptase, we can have an HIV DNA also at the end. So this is in general, DNA is happening. That is, it's going DNA replication, leading to the production of RNA and also protein. And in a special case, RNA could go back to the DNA, or RNA could self-replicate also. So these things for the special cases, and but in normal, this one is in the general cases. So further, that was all about transcription. Now let's discuss about translation. That is the first stage of protein biosynthesis. So in this thing, let me check if you have any questions. Okay, so far so fine. If you have any questions, students, please feel right, because I'm just giving you the basic format of molecular biology today. in order to get the good feel about it, what is it all about, and get a strong hold on the nucleic acids, how they are acting as a genetic material, and what is the difference between your DNA and RNA. So that's the major task for today's. I'm also a bit tired speaking last 1.5 hours. Um, just give me a second. Just 1 minute pause. Yeah. So now next part is your translation. That is the first stage of your protein biosynthesis. So in this translation, messenger RNA is produced by transcription, which is decoded by the ribosomes to produce specific amino acids. So as we had discussed in that video, which later on these proteins are folded into the quaternary structure in order to produce an active protein. So translation occurs in the cell cytoplasms where the large and small subunits of ribosomes are located and bind to the mRNA. So how this translation process is done? Basically, it's done in three different compartments. That is initiation, elongation, and termination. That's how your transcription is done also, initiation, elongation, and transcript and termination. So the ribosome first facilitates, as we have discussed in that video, that it to decode your inducing binding of tRNA with complementary anticodons and sequence to the mRNA. Then the transfer RNA carries specific amino acids that are chained together into polypeptide chain as mRNA, which passes through and read by the ribosomes. So the entire ribosome mRNA complexes will bind to the outer membrane of rough endoplasmic reticulum and release a nascent protein polypeptide inside the later vesicle transport and secretion outside the cell. So here we can see this ribosome is having this initial part with transfer RNA. Alanine is being attached now. So and it's producing a protein chain of amino acids with methionine as a starter codon, glycine, leucine, valine, alanine. Then further glycine is going to come attached and this transfer RNA will be removed and basically your polypeptide chain of amino acids attached to your ribosome will be there. So in this way our amino acids will keep come and keep joining and transfer RNA will be removing, and in this direction for your 5' to 3' end on your messenger RNA we will have continuous availability of this long chain of your amino acids. So what is the genetic code of your base pairs? So that's how they are pairing. So what we do is we write UCAG on the left hand side and UCAG on the top, and we also write UCAG on the right. Yeah, UCAG, UCAG, and UCAG, 4 letters here, and then we write the names like U from here, U from there, and U from on the top. This one. So U, then U, then C, then U and A, then U and G. So like this, we will fill these 16 boxes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 boxes, and each box will correspond to specific amino acids. So like this one will be leucines, all these amino acids. This will be the valine. So remember them, these base pairs with these yellow dots here. The full one, the 5 belong to serine, proline, threonine, alanine, glycine, arginine. So you can even draw these diagrams. Sometimes they're asking some specific sequence, tell us the name of that amino acid. So you can draw in this way. Methionine, AUG is your start codon, remember that, and the three stop codons, UAA, UAG, and UGA. Yeah, so all these things matters a lot. So I think I will just stop it here because it will be too much for all of you also. If you have any questions, I'm open to your questions. I will share this PPT also with you so that you can go through them later on. Yeah, this one. So tomorrow we will continue with genomes and another part of it, various types of DNA. Continue with various things. Yeah. It's not much. It's just 70 slides more. So we will continue with that too also tomorrow. So any questions, students? If you have anything to ask, please feel free to ask me. I will be happy to answer your things. So on that note, I will wait for your further any comments or anything, um, so that we can wrap up for today's lectures, for today's workshop, and then we will see us tomorrow at 5 to 7 p.m. again for further lectures. Okay, if you have nothing to ask, so I will say thank you very much. We are done for today. Stay safe where you are, and we see us soon. Um, yeah, tomorrow at same time. Oh, thank you very much. Bye.